Okay, so I just want to refresh your memory from last week's class where we talked about DNA sequencing, and that's kind of where we ended. But I wanted to reiterate that the method of DNA sequencing was really developed because of the way that DNA is synthesized in general, right? So in the 1970s, 1975 to be exact, Fred Sanger and others in his group found a way to be able to sequence the actual base compositions of someone's genome using this technology, using the technology behind DNA synthesis. If you guys recall when I mentioned that when nucleotides get added to a piece of DNA that's being synthesized. So if you're doing a PCR reaction, you add your primers, you have your nucleotides, right? And these are what you add your nucleotides and they get extended to the 3' OH of the preceding base. So because of the fact that DNA polymerase needs to add the incoming base to this 3' OH, they took advantage of this to generate a similar nucleotide that instead of having this 3' OH, lacked that 3' OH. So instead of it being deoxyribonucleic acid like you normally have as your composition of your DNTPs, it's a dideoxy, right? So that OH group is missing. The fact that this OH group is missing, this 3' OH group is missing, you cannot add another base, okay? So then that strand gets terminated and you can determine, okay, what was that last base that was added, right? Because that's what was terminated. And to be able to do this, right, you will do a very similar PCR reaction as we discussed previously. And you have your template strand, you have a primer, right? So you need a primer always, right? So you tell the polymerase where to start. And you start adding nucleotides at the end of that primer. Remember, because this is DNA synthesis and your primer is always 5' to 3', nucleotides get added to the 3' end. You know that this top strand, which is your template strand, should be a 3' to 5' orientation, right? What is being synthesized is 5' to 3', okay? So you're adding to the 3' end, and you're gonna replicate and synthesize DNA, and the chain is gonna grow. In this reaction mix, you're gonna have 4 different reactions for your 4 different bases. And you're gonna also have this dideoxy nucleotide that's going to be added. And randomly, as the reaction replication is happening, or DNA synthesis is happening, you're gonna have sometimes this dideoxynucleotide, which is either circled in green or blue or pink or red here, is going to terminate, okay? And it's in a test tube, you're having multiple reactions, it's random chance, you're going to have different size or different length of molecules, okay, in these pools. And once the reaction is over, you can then run a gel of each of these 4 different reaction products to see, okay, what has been synthesized. The gel that you're going to be running is a little bit different from agarose gels we discussed, right? So this is a polyacrylamide gel. It actually allows for you to separate single base pair nucleotides, right? So the resolution is much higher. So you can actually separate one base to another. Agarose gels that we've talked about previously, when you're looking at the size of your DNA fragment, you're unable to do this, okay? This doesn't have the type of resolution for you to see single base pair resolution. And this method can synthesize very efficiently, 600 to 800 base pair sequences at a time. And when you run this gel with your different compositions of the nucleotides, you know the smallest fragment, as we discussed, always runs faster. So that's going to be the smallest fragment, and that's going to be to the bottom. And here, if you look in these pools, you can see different ranges, right? So 22 nucleotides is the smallest. 33 nucleotides is the largest, right? So your first nucleotide, you know, okay, it's only an A, okay? So as you go further along, you can see the different lengths of the different DNA fragments that you see. And you can read this, right? So your primer is here, your smallest base is A, so you know that this has to be the first nucleotide that gets added. So this is the first sequence of DNA. And you will read from the bottom to the top, again, 5' to 3'. This is your sequence, okay? And because this is your sequence, if I was to ask you, for example, in an exam, if this is the sequence that you've sequenced, what is the sequence of the template strand? This is the sequence of the template strand. This is what you synthesize, right? You can see that they're matching. However, if I say, what is the sequence of the template strand in the 5' to 3' orientation, this is 3' to 5', right? Because your primer is 5' to 3'. So you will now need to, A, know what the template strand is, and flip this like you would do for primer design to read this in the 5' to 3' orientation and write that sequence that way, okay? Because as I mentioned, 99.9% of the time when you see a sequence being given or being read, it's often annotated as the 5' to 3' orientation, because that's really important to know what the orientation is. And this is just a graph showing very similar process as I just showed you in the previous slide, right? So you have your template strand, you have a primer. This is just part of a primer for ease of the graph, right? But primers, remember, they're usually 16 to 20 something base pairs long. So you have your primer, you will add your excess of normal nucleotides, and then you're gonna have your dideoxynucleotide, each one in each of the tube. The reaction gets terminated whenever it gets incorporated. And then you can run it in a gel, read your sequence 5' to 3', right? That's your sequence that you just made. This is your template strand, okay? The way that these gels were read, as I mentioned, you're using a polyacrylamide gel, which really tells you a single base pair resolution. The way that this was done previously is that you had these polyacrylamide gels and someone would just go through, right, with a pen or a pencil like you see here for Fred Sanger, looking at this film and actually writing down the sequence. So it was incredibly tedious, right, to be able to do this. So you start from the bottommost sequence and you just read the basis. So you can imagine there could be also room for error doing it this way because it's human error, right? You're reading this by eye. But it was done this way when I was an undergraduate student. It doesn't mean that I'm super old, but when I was an undergraduate student, we still did a lot of these gels, these sequencing gels, where we actually had to read the sequence for some experiments that we were working with. So, but it's incredibly tedious to be able to do this. And you really, you have your 4 bases, you run it on a gel, and you just read the sequence, and that is what your sequence will be, right? The difference between this and a regular agarose gel, besides the fact that you can get single base pair resolution, is that if you recall in the beginning, I said that you're also adding a radioisotope to this base, right? So you can actually detect it, and you can detect very small amounts. That's what a probe or radioactivity allows you to do. And you can see here on this gel, if you just look at the larger part of this gel, you can see you're starting, the first base is actually, should be C, but I excluded it here. But you can see that the first base is C because it's smallest, but this is reading from this A, and you have a string of A's, right? And there's nothing else in these lanes. And then the next base over is a C, and then 2 G's, right, as you're seeing. So you kind of read the sequence that way. So someone asked if, how is it that it terminates at that spot instead of continuing? It terminates because you can't, once you add that dideoxy, remember there's no OH at that 3' end, so you cannot add another base. So DNA polymerase cannot do anything, right? So the reaction just stalls and stops. Yeah, and that's the beauty of this technique, right? Because it allows you to say, okay, we need to stop and now read the sequence that we have. Okay, otherwise it will just keep extending and extending and extending until you run out of polymerase or you run out of dNTPs, which happens at some point. So that's incredibly tedious, right? But you can imagine it's, if you want to be able to sequence very large genome sequences, such as the human genome, right? We have about 3 billion base pairs within our genome. Going through and systematically doing this traditional way is a little bit difficult. And one of the modifications to this technique was that then they discovered that you can actually use a fluorescent label instead of the radioactive label. You can use a fluorescent label and attach it to those dideoxy molecules. Okay, so again, manual sequencing is super powerful, and it's still being, it was still used for a long time after that. But now with this tagging these dideoxynucleotides with a fluorescent molecule, you can use a machine to quickly scan through those fluorescent molecules, and depending on the color signal that you get, you know what base it is, right? And this is being transferred to a machine, and the machine just kind of prints out, okay, you had a peak of green, so that must mean that it's an A, right? So it kind of gives you a readout by just measuring that fluorescence within your tube. The way that this works is that you have your 4 bases. In the regular traditional manual method, you would run a gel with 4 independent wells or columns. Here you do this reaction as before with your fluorescent label, and then you mix everything together in one tube. This machine will then look through this tube and just read the different signal that's there. And depending on the signal that you get, it will just tell you what the bases are. So this has really made it much easier to sequence large genomes, and this is what's being used today using these fluorescent label. So if you're working in a lab and you send your DNA to get sequenced, you get back something that looks like this, right? Where you have the sequence and it shows you the peak of the fluorescence. It was also incredibly expensive, right, to be able to do this. So, back in the day when they sequenced in the early 2000s, when they were sequencing the human genome, it actually, do you guys have any idea how much it would have possibly cost to, I guess in the early 2000s, to sequence the human genome? 2.3 million. Yeah, a little bit
Billions?
higher. Pardon?
Billions?
Billions, right? So it cost $1 billion, give or take. And there were many scientists around the world working on this effort. It took them 13 years to get the full genome sequence, or a draft of the genome sequence. Of course, later on, you had to kind of fix a lot of pieces that were missing. But when they completed it in 2003, it was, it was $1 billion, right? Do you know how much it costs now to actually sequence the human genome and how long it might take? Probably a lot less, $50, $100, okay. And free, well, depending. So it costs now today about $1,000, depending on the technology you use or the facility that you use. And it literally takes a day, right? So much less time, and it's much cheaper. And that's because the technology has improved significantly. And you have these different platforms now that are able to sequence much faster, because what they do, instead of producing those very long reads, the 600 to 800 base pairs that the Sanger technology is based on, they do much shorter reads. So it's much faster and it's much cheaper, okay? So you can get usually 25 to 35 bases or 200 to 300 bases, depending on the method that you use. So these are all the different companies and platforms. They use different chemistries for the sequencing, but the idea is the same, okay? And these different technologies as well are based on the assumption that you already have some idea about some of the genome that's present, right? Because if you don't have any genome sequencing, let's say you found an organism, somewhere, it's a new organism, there's no information available for this genome sequence, you can use one of these technologies, but then you have to assemble that, right? So sometimes it's difficult, and it's difficult to be able to assemble random sequences, because how do you know which pieces go together, right? So there's some considerations that need to be made in order to really be able to do this, okay? You can sequence a human genome very cheaply right now for any person because you already have a fully sequenced genome from 2003 that you can use as a guide to help you to assemble the new genome sequences now, yeah? So yeah, and then different platforms, different machinery. The machine that was used for Sanger sequencing was a huge machine at the time. Now you have different instruments that you can use, all the way to what we have an instrument which is a nanopore technology. It costs $1,000. You can get lots of reads. And you can kind of get everything done within 6 hours. It's really small. You can put it in your pocket and walk around with it, right? So technology has really increased. Depending on what you're doing, depending on the experiment you're doing, what information you're trying to get, you have to take into account, you know, of course, advantages and disadvantages. You know, there might be some errors, you might have some mistakes in the DNA sequence. You take into account a lot of different information for this to work. And one of the ways that this is, I'm not going to explain all different technologies, but one of the ones that is very popular simply because Illumina is located in San Diego, right? And they do a lot of our large-scale genome sequences that we readily use. So the idea behind Illumina sequencing, right, is high throughput sequencing, that you're sequencing large amounts of DNA, very short reads. The idea, again, is very similar to the Sanger sequencing technology, right? So what they do is they attach short pieces of DNA, right? So fragment, you isolate your DNA, fragment your DNA, blot it on a solid surface, right? And then you try to amplify or synthesize those spots of DNA. By doing a PCR reaction, right, you're adding all of the components, right? So each DNA is amplified in those tiny patches on the surface. And then these patches are sequenced together by extending them one nucleotide at a time, right? Using the same idea, fluorescent chain termination nucleotide, as Sanger developed, okay? Your reaction is the same. Everything gets synthesized on, let's say, on this solid surface, and then it's scanned into a machine, and the machine detects those different fluorescent probe like you have. What makes this a little bit fast is because you can easily remove the block that you added to block that deoxynucleotide, okay? So you're adding this dideoxynucleotide where you don't have that 3' available. It has a fluorescent probe. It gets incorporated. The machine takes a photo of that color, right? So you have a color probe. And then you can just add a chemical, remove this block, remove this fluorescent probe so that the DNA continues to be synthesized. So you can do this over and over again in the same pool, right? And just keeps extending it. And that's why it's much faster, okay? But you also can only do this for short amounts, for short reads at a time. So the average is about 35 nucleotides long, okay? This is really what the platform looks like. This is actually an actual real fluorescent slide that you're looking at, the different colors. You know what the different color means because you added the fluorescent tag to each of those 4 nucleotides. You know which one represents green, blue, etc. And you can get the reading. It's great, sequenced very well. It's highly commonly used. UCR currently have several of these Illumina type machines available. So, and also the Sanger sequencing machine. So for small projects, like if we just want to sequence a plasmid or a small fragment of our DNA, we use the Sanger technology because you get really long reads. And if you're, you know, most of your genes are usually pretty long and you just want to see if you made the correct clone, you would use that Sanger sequencing technology here. You can also send your samples to an external company. The prices are negotiable. It costs about $4 to $5 per nucleotide. No, $4 to $5 per sequencing reaction. So depending on what you want to do, that's what you're going to pay. And you also have, if you want to sequence the whole genome, or you want to do, we haven't spoken about transcription yet, but if you want to look and see all of the genes that are expressed in the genome, you can also use this Illumina version of this Illumina sequencing technology, which we'll discuss later. But that's how the advent of sequencing technologies has enabled us to really understand what's in the genome, what are the sequences, and allowed us to actually compare between different organisms, all the different genomes that are available. So the cost has gone down significantly, you know, where we started to where we are now, and way more efficient from this process. Someone asked which, you don't need to know the difference in between the machines. I mean, you just need to know the difference between the sequencing technology, like whether it's Sanger, which gives you long read, versus Illumina sequencing or high throughput sequencing, which gives you much shorter reads. And which one is short and which one is long read. I mean, the long and short reads, it just depends on what you want. Sanger sequencing, if you go back to the slide with the Sanger sequencing, you'll see that you know, the reads that you get is 600 to 800 base pairs compared to if you're using any kind of the second generation or next generation sequencing technologies, where the reads are much smaller. Yeah. So just to summarize the concept of cloning before we start talking about other DNA replication, right? When you're thinking about everything we've spoken about so far over the last, I guess, 2 to 3 lectures, or 2 to 3 lecture material, you want to think about what do you need, right, in order to be able to successfully perform a cloning reaction, right? What do you need? You need 2 things, right, to be able to say if you're going to clone a piece of DNA into your plasmid, right, you need 2 things. So I'll help you to start here. So you need a vector and an insert, right? What are some of the types of vectors, right? You want to think about that. What are some of the types of the vectors that you might be interested in?
Virus and plasmids.
Exactly, right? So you plasmids, your phages, right? The bacteriophage, which are those in infect viruses, or you can have a eukaryotic expression vector, okay? What types of inserts can you have? This can vary depending on what you're interested in, right? So maybe you want to clone a DNA fragment, maybe you want to clone a gene, right? Depends on the question that's being asked. How might you generate these inserts, right? What are 2 ways maybe that you can generate these inserts?
PCR.
Exactly, right? So you can generate these inserts by either PCR, you can do restriction enzymes where you digest and fragment your DNA, or you can do RT-PCR, right? So you're looking at a gene that's expressed, right? And how would you visualize, after you've done your PCR, how would you visualize that you have the correct product?
We run the gel and see the size of the DNA.
Exactly, right? So you run the gel, you have the aqueous gel, so you know that it's great. Everything looks fine. So then how would you generate your vector insert fusion? You have your product, your insert, and now you've decided that you chose your vector. So you're going to use a plasmid, and now you have your insert from your PCR, which looks good because you ran the gel. How would you generate that vector insert fusion?
By cloning, put it back in the bacteria and.
Okay, yeah, exactly, right? So you clone it, right? So you use a restriction enzyme, right? You digest it, you ligate it, and you can do some kind of restriction mapping after that to see whether or not it's in the correct orientation, right? But this is actually after the fact, you know what restriction enzymes you can use to do this, right? Because you need to know which enzymes you can use to be able to do these restriction enzyme digest and ligation to get them together. So say you have your plasmid with your insert, it looks good. Now you want to put it into the host, which usually is bacteria. How would you get it into the host? How are you going to get it into the bacteria? Yeah, you'll shock it. And what is that technique that's shocking it called?
Transformation.
Right, so transformation, right? Or in that case, you can say transformation, and the shocking is really, pardon?
Electroporation?
Yeah, exactly. So electroporation is one method of transformation, right? So you'll do transformation, and you can use either chemical method, you can use electroporation, right? So just knowing, particularly those 2 methods are important, okay? So then once you have that, how do you identify that you have a successful transformation?
A marker gene.
Yeah, exactly, right? So you have a marker gene, and the marker gene, but what's the difference between your marker gene and your, what's the difference between your selectable marker and also your the antibiotic? So the selectable marker is the antibiotic resistance gene, but what's the difference between those two markers, your marker gene and also your screening marker? So what's the difference between the selectable marker and the screening marker?
Isn't it the fluorescence that's added?
Yeah, so the screening marker will have, that's the LAC-Z one, right? Where you're looking for blue-white color, right? So you're screening the colonies that are successfully transformed, and you know that they're successfully transformed because your selectable marker works, right? So your selectable marker, which is your antibiotic resistance gene, tells you that your transformation is successful, right? So you have these different genes, and so your selectable marker says it's been transformed. Your marker gene, which is LAC-Z, right? That will tell you, okay, yes, I have it transformed. These are the colonies, but does it contain my insert, right? So that's how you know whether or not something has been successfully done. And that's pretty much all of it, right? After you have your sequence, right? Your plasmid, you can then sequence it and check to see whether or not, does it look great or not? Yeah. So just think about these steps from everything. And this is kind of how I think it's good to put everything together. Like, what do you need? What are you trying to do? And how do you get to that final product of having your sequence piece of DNA to say, okay, everything looks good.
Professor,
Right?
would you
Yes.
say then that this kind of like flow chart is kind of a good idea of what to go by for like the exams or the tests? That way we kind of have an idea of looking at everything and where to go. And
Yeah, so I think you can use, I think you could use this flow chart to start to kind of put everything in place, especially the part where the cloning, right, and the vectors and restriction mapping. I will give you guys a study guide as well, which will have more information, right? So you really want to think about, I think this really is helpful for the whole cloning, because this is actually what you do in a real life situation, right? You say, okay, I want to clone something. What do I need to clone it, right? And you really go through a lot of these steps. What's different is depending on whether you're going to do restriction enzymes versus a PCR. Nowadays, everyone just does a PCR for cloning to generate your insert, okay? But everything else is real life scenarios. So if I was to ask you, okay, here's a gene sequence, you want to clone it into this particular vector, how would you go about doing it? And specifically what steps, right? So I'll be very specific about what steps I want you to talk about. Yeah, what do you do? But I think this is a good roadmap to kind of start putting everything together. Okay. So let's just quickly go over before we get into DNA replication, because I didn't get to it in the last class. I had this practice kind of homework, extra credit question listed on iLearn for you guys to go over. We didn't get to it, so I know that some people might have attempted it and had questions about it. So I want to go over this, because these are type of questions you would also see in the exam, right? So if you have a DNA fragment that you would like to clone by PCR, you need to design the primer, right? And this can be given to you, right? Someone tells you, okay, I have a gene, here's my gene sequence, design the primers to do the PCR to clone this gene. One of the things that you want to think about beforehand when you do this is that you know that this is the sequence, and I wrote the sequence and I just wrote a bunch of dots in between just because it's a long sequence. So this is the beginning and this is the end of the sequence. This is not two individual sequences, right? So this is one sequence. I just didn't put all of the information in between. And then I would ask you which of the following pairs of primers will direct the amplification of this gene, or I'll ask you to write the primer that will amplify this gene, right? So it could be either of two ways, yeah. And the first thing you want to do when you attempt such a question is always think about orientation, right? This is really important for everything that you do related to sequences. If you are given a sequence, 99.9% of the time, if there's no annotations written on this sequence, it has to be 5' to 3', right? This is just a given. Yeah, this is acceptable. This is what's acceptable in the field, unless someone says otherwise. Yeah. So I would recommend you write the orientation on the sequence. DNA does not exist as a single strand, so you need to write the other strand. This is absolutely important, right? DNA is always double-stranded, right? Unless you make it single-stranded, but DNA does not exist as a single-stranded molecule, right? It's a double-stranded molecule. So, you know, your first strand is 5' to 3', your bottom strand has to be 3' to 5', right? It's complementary to the top strand. So that's what I would suggest that you do first to attempt these type of questions. The next thing you want to do is, okay, when we design primers, you need a forward primer, right? That goes from where you want to start, and you need a reverse primer, tells you where you need to end, okay? So if you were to have a forward primer that starts here, and I said the primer needs to be written 5' to 3' orientation. So if you have a 5' to 3' orientation for your forward primer, where does this primer anneal? Where would this primer bind to? So you're starting here. This is what you want to amplify. Where would the exactly, right? The primer needs to bind to the bottom strand, right? Because this is 5', this is 3'. It can only bind here, right? To this bottom strand, right? Because the basis has to be complementary, right? So this A has to match with this T. So a general rule is that if you have a double-stranded molecule, you have the orientation written, the forward primer is always going to be similar to this top strand, this 5' to 3' strand, because it has to anneal to the bottom strand. General rule, always the case, right? That makes sense because these two strands are going to separate when you do the denaturation, and then your primer is going to anneal to this bottom strand. So now you need something to anneal to this top strand, right? Because you have two strands, right? Forward primer anneals here. So that means that your next primer have to anneal to this top strand. It's reverse, right? It needs to be written 5' to 3', right? So your 5' is going to start here, right? So your sequence of your primer is going to be going in this direction, right? This is the complementary to this top strand, right? This is the sequence. But this needs to be written from this direction. So then you just flip it, right? So you start T, A, G, right? So this is your sequence, right? And this primer then can anneal to this top strand. Is that clear?
Yes.
Okay. And someone mentioned about the coding versus the template strand. And doesn't really matter whether it's the coding. If the question specifically asks, like in this question, at the time when I wrote this question, we assume that this is because a coding strand just means it's the gene, and a gene starts with ATG and ends with a stop codon. So this is always the coding strand. So don't think too much about whether it's the coding or the template strand for these type of experiment. Coding just means it's a strand that codes for your gene, and that's usually the top strand, and that's usually the 5' to 3' strand, because that's how the DNA is written, right? ATG to your stop codon. So don't worry too much about that. Just worry more about where you're starting, where you're ending, where your primers annealing, okay? Because that's how you're going to.
So we're gonna always be given the coding strand.
You're always going to be given the coding strand, or you're always going to be given the top strand, right? You can assume that the top strand is 5' to 3' orientation, unless I say otherwise. Yeah.
Okay.
Okay.
And before we go on, can you explain again the purpose of the site-directed mutagenesis? That's
This
called, I think.
this the site directed, was this the one for, this was a few
Mm.
lectures ago?
Yeah, like, yeah, I think the lecture before last lecture. Yeah.
Yeah, so maybe let me get through these lectures, because that's a really different topic with the mutagenesis, where you're trying to
Because I mean, like, you were
it-
giving like summary of everything we learned, right? So
Sorry?
you were giving summary of everything we learned. So that's
Oh
why
no,
would
so I
like
just want to give you, yes, I don't want to go into detail about every single step in terms of the cloning. I mean, so the site-directed mutagenesis is a little bit different, right? That's just another way, another method of PCR, another way you can use PCR, right, to do something, right? That's what that was about, right? So we can talk more about it either during office hours or if we have time after, so that I don't stop here to talk about that, yeah? But that's just another technology that's just another way for you to use the PCR technology to study a particular gene function, right? And in that case, it was really trying to change a protein, you know, from one codon to another codon to see whether or not it was important. Yeah. So the other question that I asked about, which you will see is more about restriction enzyme, is we had these questions where you have two commonly used restriction enzymes. Initially, when I wrote this question, I had 3 and I removed 2. So if you listed 3, I just graded the 2 that was correct, which only 2 of them should have been correct. So you have 2 pairs of commonly used restriction enzymes, and you need to figure out the compatible ends, right? And the answer would have been BCL1 in this case, and BAMH1, or CLA1 and NAR1, okay? And the way you go about these questions, because I know that some of these questions can come up in your MCAT exams, right? You want to go about these questions thinking more in terms of where are the restriction enzymes cutting, right? And what type of overhang you're going to have to see whether or not it's compatible. So again, when a sequence is listed, you always want to write the orientation, and you also want to write the strand, the other strand, right? It's not a single-stranded molecule unless we're talking about RNA. It's often, you know, written as a double-stranded molecule. So if you're given the enzyme sequence, in this case you get BCL1, it has to be 5' to 3'. You just write the other strand, right? And you do it for both of them or for all of them, right? Just so you can have an idea of where your enzyme is going to actually cleave, right? So BCL1, why are these two compatible? Because BCL1 will cleave here, right, on the top strand. It will cleave here on the bottom strand. So this is going to be your overhang. BAMH1, yes, the sequence is different, but BAMH1 cleaves here. It cleaves here, and this is your overhang. So these two are compatible, right? And you do this for all the other ones just to see, yeah? So that's kind of how you want to address these questions. Okay?
Professor?
Yes.
Yeah, I tried to do the CLAI plus NARL one, but I couldn't get the, like how we did this one right now for BC2 and BAMHI. I couldn't get it.
Yeah, let me try to quickly see if I can. Okay, so the other one is this CLA1, right? And NAR1, which you couldn't get, right? So again, you wanna, I'm gonna try to write this, right? So bear with me. Right, the other strand is right. So this is CLA1. This enzyme cuts here, and this enzyme will, the bottom strand will cut here, right? So you have, this is your overhang, your GC. So NAR1 is exactly the same, right? So NAR1, you have G. right? NAR1 also cuts here and cuts here. So what do you have? You have the CG, the GC, right? So these are your overhangs and they're compatible.
So when you say compatible, then we're looking at the two different
Enzymes
restriction
that have
enzymes.
the same overhang, because
And we want to make, and that's what we're doing then, is we want to
then
write
to
it and
one
make sure
together
that the
and mix
over.
together. Yeah.
So then we just write it out and make sure the overhangs actually would line up and match the way they're supposed to.
Right.
Okay.
So if you digest with this enzyme, then your overhang should be able to be annealed in a vector that was digested with BCL1, because this is the overhang. So yours will match this. Yeah.
Can you show the overhang of PSTL?
Which one?
The PSTL or this pistol, I think. Pistol?
With this one?
Yeah, this one.
I mean, this one is going to be the same, right? Because it's going to cut here, and this overhang is going to, the other enzyme to the bottom strand is going to cut here. So this is going to be your overhang.
Oh, okay.
Yeah. And you can see that this doesn't match up with E. coli 1, so you know that these two are out, right? Okay.
So then it's just a process of elimination of figuring out which ones work. There's no real way to tell,
I mean,
one works with another, or.
The way to tell is that you have to write them all out, right? Also, if you look at this GATC, neither PST1 or E. coli 1 matches this or this, so they don't go anywhere, right?
Professor?
If you went through all of these and you see which ones have the compatible ends, if say you decide to do PST next, this is the overhang for PST. This doesn't match any of these other overhangs, so you have to eliminate.
I'm sorry, what is the purpose of of having the compatible cohesive ends? Like, I just kind of.
For cloning, so let's say you remember, so if you want to clone your insert into a plasmid, sometimes you cannot use the same enzyme. So you cannot maybe you cannot digest your insert with BAMH1 and clone it into a vector that's digested with BAMH1. So you need to use two different enzymes, okay? So you need to know which two enzymes then in that case will have compatible ends. So let's say you're going to clone a gene, and your vector that you have, the restriction sites available in that vector is BAMH1, okay? In your gene, you have a BAMH1 site. You cannot use it because you don't want to cut inside your gene. You want to cut outside of your gene, right? So you have to use something else. So then you have to look to see what else can I use, and then look to see whether or not those sites are compatible with BAMH1. Yeah.
Okay, thank you.
Okay. Let me get out of this. Okay. So yeah. So I mean, again, if you guys have more questions, again, you can either office hours or Israel in discussion sessions, or his office hours should be able to help to clarify some of these. Yeah. But really, you have to kind of just work through them, write the orientation, write the order strand, indicate where they're going to cut, and then see what the overhang is and see whether or not it's matching. Okay. Oh, so let me get rid of this now. Sorry. Eraser. It seems like it stays there. So now we're switching gears and talking about DNA replication. Some of the concepts of DNA replication is very similar actually to what we just talked about. So because DNA replication is kind of synthesizing DNA, right? So you're kind of replicating the DNA, and polymerase is doing that, and you're adding nucleotides and extending that new strand of DNA that's being replicated, okay? So some of the questions that you want to think about as we go through this next half of the lecture for DNA replication is, do the old DNA strands pair with the new or the old DNA, right? How is DNA replicated? What are some of the proteins or important proteins that are involved and required? How does the cell know where to begin DNA replication, right? Do you just randomly start replicating a piece of DNA for no reason? And does replication go in one direction or both directions? It's DNA replication. You're using an enzyme. You're adding bases. Is it, how accurate is it? What are the chances of you making a mistake, and how do you fix those mistakes, right? Because your DNA is double-stranded, you need to, you're not doing a PCR reaction, right? Where you can stick it in a test tube, you can stick it in a machine, and you can denature the DNA and separate the strands, right? This is in the cell, right? So you need to unwind the DNA. And unwinding the DNA is like, imagine that you're pulling a zipper apart. So you're gonna have supercoiling and tangling. How does DNA replication deal with that? How does the proteins deal with that tangling and supercoiling in the genome? Okay, so we have, as I mentioned, about 3,000 million base pairs or 3 billion base pairs of DNA in our genome. You need to copy that every time the cell divides, right? And how many cells do you guys think we have in our body? Like just a rough estimate. Zillions, trillions, something. Yeah, so we have trillions. So we have about 30 something trillion cells in our body. And you can imagine that all of those cells need to be replicated at some point or the other, right? So it needs to really be a precise process and ways to fix a problem if there's a problem or an error, right? Because you don't want to have detrimental effects, right? You don't want to have a problem within your cells like cell death or other types of mutations that might be lethal to the organism. So in the beginning, once, we went through all of this process where we talked about how DNA was discovered, how we knew what the genetic component was that was being transformed, right? There was a lot of work done trying to understand how is DNA replicated within the cells, right? If cells are going to multiply, you're going to give rise to new cells. What is the mechanism of this DNA replication? So the initial idea was that we could only have 3 possible mechanisms, right, for this to actually happen, right? Because DNA is double helix, so you know that you have a double strand. The 3 mechanisms that were proposed at that time was either semi-conservative, right, where you have 2 of the original strands get separated. Once they separate, you have a new strand, right? So what you have in the end is an old strand and a new strand, right? So it's semi-conservative because you're kind of only conserving one of the strands, right? That's present in this new replicon. Conservative, meaning that what you started with, right, your original double strand, you also have this double strand after the DNA gets replicated, and then you have a new set of daughter strands present as well. The other option is dispersive, right, where you start with this double strand, and somehow, instead of the strands being separated, they kind of fragments of this strand anneal to each other, right? So pieces of it gets synthesized, you have an old strand, and you have another piece of new strand, and then you have an old strand, right? So this is possible. This was also a possible mechanism. We didn't know at the time which of these 3 mechanisms. So a group that was, studying DNA replication at the time decided to do an experiment to figure out what is the possible mechanism of these 3, right? This was their hypothesis, that DNA is being replicated in. And these experiments were done by Matthew Meselson and Franklin Stahl. So it's usually referred to as the Meselson-Stahl experiment, as you'll see in the next slide. And they just did a classic experiments using centrifugation to separate the strands of DNA to see whether or not they can figure out which of these 3 hypotheses might be true. So the idea behind this experiment is that you will label the DNA, the initial DNA, you will label with a nucleotide, okay? Nitrogen 15 isotope is a little bit more stable, so they use nitrogen 15. It's also heavier, okay? So they added this radio isotope to the DNA it's replicating, and you have it, right? So this is the original DNA that gets replicated in blue. After that replication happened, they decided, the first round of replication, or when the DNA is replicated a little bit, they decided to add another isotope, which is a little bit lighter, right? And it's more the normal state of this nitrogen isotope. So this isotope will then get incorporated into all newly synthesized DNA, right? So your initial pool, you have your DNA in the tube, you label it so you can have the old DNA labeled with this heavy isotope. All newly synthesized DNA or replicated DNA will then have this lighter isotope, or the normal N14, okay? You can then use a centrifuge because N15 is heavier than N14, right? So you can have a centrifuge and then look at the separation on the in a test tube, right? So if it's heavier, it's going to sink to the bottom. So this is the test tube. This is the bottom of the test tube you're looking at, right? So it's heavy and this is lighter. So this is more the top of the test tube. And then they use cesium chloride because cesium chloride is also like a chemical that allows you to separate. It's better than water, right? So it allows you to separate gradients in this DNA solution as well. So they did just added this into the solution and then did centrifugation to really separate the weight of the DNA, how dense the DNA is at that time. So these three hypotheses, as I mentioned, if you can imagine that it's conservative, right, which we talked about, you have the DNA is labeled, it's heavy. If it's conservative, after the first round of DNA replication, you will still have the original strand, and then you would have the newly synthesized daughter strands. So then when you separate this in a test tube by spinning, you will have two bands, right? So you will have one that's the lighter and one that's the heavier, right? This would be what's expected if it's conservative. If it's semi-conservative, what you would expect, again, you have a mixing, so original strands, each of these strands serve as a template, your new daughter strand is present. Then you would have a mixture. So instead of having light or heavy, you have a middle ground, right? So you have an intermediate, which is shown by the purple here. Similarly, if it's dispersive, you will also have the same as semi-conservative because you're mixing it, right? So you're mixing heavy and light, so you get a middle ground here, right? So because of this, these two options, right? You have to find a way now to go beyond that to say, okay, is it really semi-conservative or dispersive if you have this middle band? And that's where the next generation helps to clarify that, because you go to the next generation, right? Again, you're using these as templates, right? You're gonna have this, right? And you will synthesize when these two separate. Your blue is gonna make a new red. This red is going to make a new red, right? And the same for the other daughter strand. So then what you have is you're gonna have the middle ground, and then you're gonna have a lighter strand. However, with dispersive, because everything is mixed together, the pieces of the DNA is always gonna be fragmented together, you're gonna get this middle ground showing up again, right? Where you can't really distinguish between heavy and light here. And when they did these experiments and tracked it over generation time, what they found was that indeed, what they saw was this semi-conservative observation, okay? So they were able to rule out conservative and also dispersive. And this is an actual gel of that experiment that you're looking at here, right? So these are the isotopes that they're looking at. So the test tube that you're looking at, this is the bottom of the test tube. This is the top of the test tube. After the first generation, right, you have that middle ground when it's semi-conservative. After the second generation, right, you have that middle ground, but then you also have a lighter band, a higher less dense band, right? So it's higher, right? Because that's a newly synthesized one. And then after the 4th generation, you start getting more and more of that lighter band, right? Because you're diluting out that original band, right? So based on these observations, they were able to conclude that actually DNA is indeed semi-conservative, okay? And once they said, okay, if the DNA is semi-conservative, you have this double-stranded DNA that you need to unwind and start synthesizing or replicating. Does it continue, does both strands continuously synthesize equally? Or what happens because they have orientation, right? One is 5' to 3', the other one is 3' to 5'. So how do you synthesize that, right? So it turns out that, right, both DNA strands we know needs to be synthesized, but the process is kind of different, right? So the hypothesis is that, okay, it could be continuous, right, when both strands are separated. The 5' to 3' end, you're going to synthesize. The 3' to 5' end, you're going to continuously synthesize. The other option is that perhaps it's semi-discontinuous, right? So one of the strands is continuous and the other strand it makes parts of it, then it makes another parts, it's not continuous, right? There's gaps in between. Or it's both strands are discontinuous, right? Where pieces are synthesized and there's gaps in the middle. So they didn't know this at the time, right? So they had to do experiments to figure out how is this DNA being replicated with these 2 strands. And because of the way that bases get added to the preceding base, we knew, right? You guys knew this from all the previous lectures that DNA polymerase can only add nucleotides to the 3' end, right? So that information they had. So because of that mechanism where DNA polymerase can only add nucleotides to the 3' end, they knew that the mechanism had to be different between these 2 strands, okay? Because you can easily add here, but how do you add, how do you go in this direction and adding bases to this end, right? How do you do that, right? So what they found was that the leading strand is made continuously, as you would expect, right? 5' to 3', no problem. As you open the replication fork, it keeps extending. However, the lagging strand is made discontinuously, right? So kind of like how this one is made, yeah. Then once the lagging strand is made, you have DNA ligase, which is the enzyme we talked about when we were talking about restriction enzyme that help to seal those gaps, right? And seal those pieces of DNA together. These short pieces of DNA were usually called Okazaki fragments, right? So these fragments that are made because of the scientist Reiji Okazaki, who developed this technique, who determined that these fragments were actually happening in DNA replication. And how he was able to actually do that was by running a series of gels and looking at the radioactivity that you have for the different fragments of DNA when you had DNA ligase or not, okay? So I'm gonna try to go through this experiment and explain it pretty as simple as I can. Let me just make sure, okay. So these are 2 different experiments that he performed, right, in bacteriophage, right? So what these experiments are showing on the left, this is wild type bacteriophage, normal bacteriophage, nothing is happening. On the y-axis, you're measuring the amount of radioactivity. So radioactivity counts per million per milliliter, right, in thousands. This is just the units radioactivity you're looking at. So the higher it is, the more radioactivity you have, okay? On the x-axis, you're looking at the distance in a test tube. So you could imagine that this is a test tube, And this is the bottom of the test tube. This is the top of the test tube. So 0, top of the test tube, 4 is the bottom of the test tube. Okay. So if the heavier something is, or the longer the fragment you have, it shifts more towards the bottom of the test tube. Okay. This is exactly the same type of graph you're looking at here. The difference here is instead of having a wild type phage, you have a mutant phage. This mutant phage is mutant in the fact that the DNA ligase for this phage is not functional. So it's unable to stitch or seal gaps in DNA. It's unable to stitch anything together or to ligate anything. So it's not functional. Okay. So what he did is that he labeled the DNA with short pulses of radioactivity, right? So just to get enough of those radioactivity into those replicating DNA, okay? And by doing that, when the DNA replicates, you can measure how much radioactivity is there, right? So if the fragment is short, you have less radioactivity, right? So you can imagine you have a short fragment. If you have a long fragment, you have more radioactivity, right? Because you're gonna just give these pulses and incorporating the radioactivity, and at some point you have a lot of radioactivity. The fragment is much longer, right? So longer fragments, in this case, have way more radioactivity than a shorter fragment, okay? So he did this during DNA replication and found that in this wild type phage, whenever he gave these short pulses of DNA, that initial short pulse, like after 2 seconds, right? So that's the first set of DNA that gets replicated, right? You had these short pieces of DNA. And they just continued on, allowing this reaction to continue, and then giving pulses to see, okay, as it takes a little bit longer, maybe after 60 seconds, how much radioactivity do you have, right? And you see that it starts to go up, right, after 60 seconds. After 120 seconds, 2 minutes, you see it's even higher radioactivity. So what they suggested is that these short pieces that they started with are somehow getting longer. And the way they're getting longer is because they're being stitched together, okay? And you have a longer fragment. However, in this example, yes, after 10 seconds here, right, you have short pieces of DNA, tons of radioactivity. So that means that these short pieces of DNA are really just accumulating, but not being stitched together, right? Because they're not getting longer. They're not heavier, so they're not getting longer, right? So they're shorter, they're at the top of this tube floating around, and they're highly radioactive. So what they suggested is that, yes, they're being made, but they're not being stitched together, because if they were being stitched together, they would be towards the bottom of the tube. Okay, it will still be as high in terms of radioactivity, but it will be at the bottom of the tube. And the reason that they're not being stitched together is because you don't have ligase, a functional ligase, to be able to do that. Okay, so this was really the experiment that really showed that these pieces of DNA was being made on that lagging strand, and its ligase actually was responsible for stitching them together. Okay. And around here, this is one, you see this sharp downturn here because they didn't give a pulse here, right? So you're really looking at the readings, like really when they actually give these pulses. So at this, you have this increase, it's attaching, right? And then it starts accumulating, right? So this dip is really just because of the nature of how the experiment is. Yeah. And this is really, this is kind of normal, right? So you have the short pieces being made, they start being assembled together, so you will get a dip because you're gonna lose, as they stitch them together, you're losing some, but then as they're getting longer, you're adding more radioactivity. At all of these times, it starts increasing, right? Because these peaks are measuring how much radioactivity you have and also how heavy they are in the tube. Okay. So these were the experiments that defined that that's what's happening. Now, gosh, I keep moving this because something is in my way, sorry. So this happens because, as I mentioned, you have to be able to synthesize DNA. And the way that polymerase works, it can only synthesize DNA adding to this 3' OH, right? So your nucleotide comes in with the triphosphate, it binds to the OH, you get rid of the diphosphate, and you have this phosphodiester bond, okay? It requires, DNA replication really requires a primer with a free 3' OH, okay? So you need a primer, okay? It just cannot see the double-stranded DNA, cannot see, oh, the strand has been separated, I should just start replicating here, right? It needs to know where to start, okay? And once it has somewhere to start, then you will have a nucleotide comes in, right? DNA polymerase will recognize it, and at that base, everything goes fine, okay? So this is kind of what have to happen. Because you need that primer needs to come from somewhere in the cell. And the primer comes from an enzyme called a primase, which makes these little pieces of primers to tell RNA polymerase, you can start replicating here, okay? So as the replication fork opens, these short RNA primers are made by this enzyme primase, which is kind of like an RNA polymerase, but it's called primase. And then now DNA polymerase can now add and extend the nucleotides, replicating the DNA. The reason that this primer is RNA, not DNA, is that the assumption is because RNA is not as specific as DNA. So the matches, the base pairs are not going to be perfect. Also because it's easy, you'll have to remove this, these RNA primers need to be removed at the end of DNA replication. And it's easy for the machinery that removes this to recognize this as being RNA and not DNA, so it can easily chew it up and get rid of it. Okay, so this is kind of how the DNA polymerase knows where to start replicating this DNA. Where in the DNA does this replication happens, right? Does it happen in one direction, both directions? In this case, you have a circular plasmid, as bacterial plasmids are circular, right? You can also use radioactivity labeling to determine what is happening in terms of the direction of DNA replication. And this is exactly what's been done by Cairns. So this Cairns experiment in the 1960s is where they use radioactivity to label the replicating DNA and then look on a film or a autoradiograph to see the pattern that you're seeing of that replicating DNA, okay? So similarly, you would grow the bacteria in radioactive hydrogen-3, and then you would image it and look to see what do you see, or expose it to a film and see what do you see. And this is kind of the depiction that was seen, right? Where you had these two edges, which suggests is kind of part of the circular plasmid. And then you see this opening. And this opening is what told them that this is possibly the origin that you have. And you also see that there's a fork here, and then there's also a fork here. So that told them that DNA replication is going in this direction, and that direction, so it's bidirectional, right? But this is not true for in all organisms, right? In bacteriophage, this is not the case. It could be directional in bacteriophage. Okay. And if you imagine that you have a plasmid that's circular, and the way that you can imagine that this origin of replication happens is that you have this opening between these two strands. And imagine that you're grabbing onto this red part and just pulling it, right? So you're separating it, right? So this bottom strand, the newly formed DNA is going to be replicated, and then the strand that you have, because again, it's the one that was base pairing here, you will have the new double strand or new DNA that's being replicated here, right? And you end up with your two fully replicated molecules, okay? And this is actually what they saw. This is actual radiograph of a bacterial plasmid showing what's happening as the replication is happening. It's exactly like what you see depicted here or shown here, okay? So this is just a schematic to highlight even more. You have this single origin of replication. It looks like a theta structure, so you'll see it being referred to as a theta model of replication in this class or also in future classes. And you have this origin of replication opening showing that DNA is being replicated. So you see these new blue strands that are being made, right, as you're going away from that origin of replication. And you have your double-stranded molecules in the end. You're gonna have direction, right? So this is the strand that's going, this is a 5' in, this is your leading strand, and then this is your lagging strand. The other origin of replication, same thing is happening, right? We're going towards the replication fork, it's the leading strand. Away from the replication fork is the lagging strand. And this is what's happening in DNA replication. And so you know now it's bidirectional. In order for this to really, this was really shown is that, you know, you have this image which I showed you where they demonstrated that DNA is bidirectional, right? You have these two replication forks here. Radioactivity, the minute that the DNA, that bubble or that origin of replication separates, the DNA gets incorporated with a very low pulse of radioactivity, right? So it'll continue to replicate. Then you add a higher or heavier type of, or a longer pulse, I should say, of radioactivity to really increase the intensity. So this is low, so it's a little bit weaker. Higher pulses will be much darker, right? And you can really clearly see that depending on the amount of radioactivity you're adding. This also helps to distinguish DNA replication in eukaryotes and how that might be happening. Is it happening bidirectionally as well, or is it happening directionally, right? So all I told you about was in bacteria. The same idea was done in eukaryotes. Of course, the mechanism is a little bit more complex and we're not gonna get into that. But the idea is the same, right? Eukaryotic chromosomes are linear, they're not circular, right? The bacteria chromosomes are circular. So you can see here, you have the chromosomes that line up, and you can see if you have kind of an origin of replication, you have in both directions DNA being replicated, right? And in this experiment, in order to show this, what they did is that they did high radioactivity first, and that's why this is brighter. And then here they had a lower activity, so it's weaker. Yeah, and this is kind of opposite to what you saw in the previous one where it was much higher here and much weaker here, okay? So you knew that, okay, you had one origin of replication and it's going outward as well, and it's bidirectional in both directions, okay? If it was just a single origin, I said if it was just a single origin, then it would be here and it would all be going, this would be brighter. And if it was directional, this would be brighter and then go weaker, so brighter, weaker, but that's not what they saw. The difference between these two graphs is that with bacteria, you can synchronize the cells, right? So all the bacterial cells are going through replication and dividing at the same time. They didn't do that here, and that's why, you know, you have different intensities. Like here, it's, you know, much brighter. Here is, you know, a little bit different. But in this experiment, they kind of synchronized the cells, so you can see nice. All of the different chromosomes, they all have kind of the same intensity, so they're dividing pretty much at the same rate. Okay, so don't get bogged down too much by the details about these two experiments, besides the fact that both experiments using different pulses of radioactivity showed that DNA replication in bacteria is bidirectional. DNA replication in eukaryotes is also bidirectional, single origin of replication. Okay. So what else is needed for DNA replications? We talked about the fact that the direction, the origin of replication, the way that the lead-in versus the lagging strand is replicated. You know that you have these little fragments that needs to be stitched together. Critical to everything is the enzyme that actually performs the synthesis, right? And that's polymerases. We talked about them in the PCR reactions and also throughout the previous lectures. Bacteria have 3 DNA polymerases, right? So there's DNA polymerase 1, 2, and 3. Polymerase 3 is the polymerase that's responsible for DNA synthesis and replication, okay? Or I should say DNA replication, it's polymerase 3. These other polymerases can also play a role in DNA synthesis, but not as efficient, okay? DNA polymerase 1 is also known to be involved in DNA synthesis as well. But polymerase 3 is the main enzyme that's important for, that's being used for DNA synthesis. It's a multi-protein complex. So within this polymerase 3, as you'll see, when we talk about transcription, not transcription, when we talk about more about DNA replication, you'll see that there's multiple components that makes up this polymerase 3 multi-protein complex. DNA polymerase 1 or DNA polymerase 2 is also involved in repair, right? DNA repair. And all 3 of these can only synthesize DNA 5' to 3', as I mentioned. They also have exonuclease activity. So exo meaning on the outside compared to endonucleases. We were talking about those enzymes that can work within the DNA, like restriction enzymes. So these are exonucleases, meaning that they can function at the ends of DNA. All 3 of them are able to proofread DNA or repair DNA 3' to 5', so at the 3' end, right, of the DNA. But DNA polymerase 1 can also do 5' to 3'. And this is important later on when we need to do, when you see you have to actually fix and repair gaps in the DNA, particularly on the lagging strand. Okay, so DNA polymerase have the ability to do both ends of the DNA. In addition to this activity, as I mentioned, only can make DNA. They all require dNTPs to be able to function with a primer that have a 3' OH. Otherwise, it does not work. Okay, so it just cannot function without having that primer available to add the incoming base and extend the DNA. The way structurally that happens as well is that DNA polymerase recognizes and utilizes base pairing rules, right? It knows which base is supposed to be sealed together or added together based upon the structure and also the topology of the double strand, right, within the cell, and also the spacing between bases, right? So if you have a strand of DNA, you know that you have a G here, you know that you need to add a T here. Similarly, if you have A and you want to add a T, for example, DNA polymerase will recognize the preceding base and knows what needs to be added. If for some reason this incorrect base gets added, so you have an A and you're going to be adding a T here, it will change the topology of this structure. DNA polymerase recognizes it and will not catalyze that reaction of adding that base. Okay, so they know it's a mistake. The DNA polymerase will stall, and then you need to repair it before it continues. Okay, and we'll talk more about, we'll try to get through the remaining slides where we talk about the repair mechanism and how DNA polymerase helps to repair kind of these mistakes when it happens. But you can imagine these mistakes do happen, you know, in the test tube. if you imagine that in a test tube, if you were going to be synthesizing this in the test tube, you can have, you know, one mistake, one base pairing mistake in 100,000, right? So, you know, that's it's kind of frequent, right? So you want to really be able to repair these mistakes as they happen. And we'll talk about how that's done in the next set of lectures. But I'll stop there and take questions if you guys have any questions before we continue, before I shut off. Yeah.
Which parts of the DNA replication here, going through lecture 4, should we really focus on? Do we need to worry so much about like some of the smaller details, or kind of worry about like the bigger details, you know, the Ozaki fragments, what was done to identify how the DNA replicated, things like that?
So with those Okazaki fragments, you really need to just know that this is important for, or this was the way that the lagging strand was shown to be replicated, right? So you need to know what they are, right? I don't think that I usually ask to describe the experiment that identify them. I think it's more important for you to know that the 2 strands of DNA are both synthesized, but just using a different mechanism. And the reason that that difference exists is because DNA polymerase can only extend on the 3' end, right, which you don't have. So that means that you have to have these short fragments being made 5' to 3'. And we'll talk about that also, I think, later down in this lecture. So every time the fork opens, right, a primer gets added beforehand, synthesized, added, synthesized, right? So you have to have these pieces. And that's really important because of the way that DNA polymerase function, and you need to replicate both strands of DNA. So I think for that part, you need to know, you need to understand the polymerases, the different types of polymerases, right? Also understanding the fact that DNA is semi-conservative, right? So I could provide a type of an experiment to ask you if it was conservative versus semi-conservative, what would you expect after the first round of DNA replication, right? So just understanding these basic mechanisms of DNA replication is important. Yeah.
Okay, thank you.